Hi, everybody. I think we should go ahead and get started because we're going to have two speakers tonight. And before we dig into that, just want to remind folks we'll have project updates next week. And the following week, we'll have Leon Chen, who will be speaking about commercialization and pitching your project. So this will be a good thing. So tonight, we are very fortunate to have two longtime SPARK advisors who are going to be speaking with us. And it's great to have them both here. First speaker is going to be Dr. Bill Fitch. And Bill is an adjunct professor at University of California at Santa Cruz and at Stanford. He currently consults in drug metabolism, bioanalysis, and mass spec. Does a lot of work here at Stanford, helping move programs forward. Bill received his bachelor's at the University of Michigan, rough last week in December. He got his PhD here at Stanford, did his postdoc stints at Yale. Is he? All right. So postdocs at Yale and did synthetic chemistry at Syntex and clinical mass spectrometry here at Stanford, and spent 31 years in biopharma in industry, worked on applications of mass spec in various areas, including environmental analysis, agriculture, chemical discovery, combinatorial chemistry, and also pharmaceutical discovery. Bill has multiple publications, and he has been a tremendous friend and advisor to SPARK, so we are very happy to have him.
Well, thanks for inviting me, Kevin, and nice to see a lot of friends out in the audience. So I'm going to talk about how you identify metabolites in drug discovery and development. And I've been having discussions with Kevin over the last year on how Stanford is going to put together its drug discovery efforts, particularly in the areas of metabolism and DMPK project management. And on a longer scale, you know, what should Stanford's position be on educating people in these areas that there aren't a lot of education programs in the United States anymore. So let's concentrate on metabolism. And this is a slide I got from Roche. And all my slides, you'll see a little hint to myself down in the corner where I got the slide from. So this was a timeline chart that we put together back when I was working at Roche in the 2000s. And I don't know if all the acronyms are still in use, but you know, lead identification, lead optimization up through marketing, doing soft spot analysis, investigative metabolite identification, profiling in vitro and in vivo and carbon-14 studies, all the way through clinical and the final carbon-14 study done in man. And I'm going to concentrate on two areas here. I'm going to say just briefly on some early metabolite ID work that you would do, soft spot analysis, and then finally describe a case study that I recently completed for a molecule that's just going into the marketplace. So this is a screening cascade that we had back at Roche for some kinase or another. And it shows how early metabolite ID might fit into a lead discovery program, starting with an enzyme assay. Maybe the chemists are doing some in silico calculations on their molecules. But when a molecule looks pretty good, you put it into some in vitro assays like metabolic stability and permeability. Maybe you start doing solubility. But before you go into single dose PK and in vivo efficacy studies, it might be nice to do some, if I can figure out the pointer, let's see, there it is. Yeah, that sort of works. Some high throughput metabolite ID. And what that is just a quick feedback to the chemists on what are the spots in the molecule that's causing the metabolic instability. So I'm going to illustrate this with some slides I took from my good friend Shicheng Mao. I don't see him here today. He comes here a lot. He put these slides together 20 years ago, and I can't do better than them. So I just keep using them whenever I'm describing how you use mass spectrometry in metabolite structure determination. So the fundamental experiment would be to take human liver microsomes and look at a 0-time sample and a 1-hour incubated sample. We illustrate with this drug buspirone. And then we do the LCMS of the 2 samples after sample prep. And the one at time 0 only shows 1 peak, which is due to the unchanged drug. And you can see it's molecular ion. And now we have accurate mass spectrometers, so we can see those out to 4 decimal points and really identify the molecular formula of any metabolite. And then we look at the metabolized sample. So there's a bunch of new peaks here. What can we learn about them? Well, the mass spectrometer tells you the molecular ion and hence the molecular formula for each one of those metabolites. And very often, a 386 molecular weight or molecular ion drug will give a 402 molecular species on metabolism. That's the addition of 16 or 1 oxygen, the most common reaction in drug metabolism, probably due to cytochrome P450. And there's a number of those. There's a number of isomers. There's maybe one that's got 2 oxygens added. There's one that's lost a few carbon atoms. But then we go and do the MS experiment. And this is illustrated just on one of those metabolites. You have to do this for each one of those peaks. But we pick M4, and we do the MS spectrum of the parent drug. And we can interpret these ions at M over Z, 122, 150, 222. And a scientist expert in the field can draw these little lines that show where the fragmentations are happening. And I'm not going to go into the details of mass spectrometry and MS. Just take my word. It's a way to take molecules, isolate them in the mass spectrometer, fragment them into pieces that give you structural information. So then we look at the same fragments in the metabolite. So we've got the 402, the plus 16 on the 386. That's easy to identify. And many of the fragments are unchanged in the metabolite. 122 stays at 122. 150 stays at 150. But some of the fragments are changed by that biotransformation mass of 16. So you put that information together with the fragmentation pattern that we figured out, and it tells you the structure. And so in this case, we know that the hydroxylation has to take place in that internal 3-carbon piece. And maybe we can limit it down a little bit by using chemical intuition on which of those metabolites would be stable. But the chemist doesn't really care about that a lot of the time. They just want to see what's the liability in their molecule when you're doing early metabolite ID. So I'm doing this currently for a couple programs at Stanford. One of the scientists in the engineering department is working on Paxlovid analogs. And in the Jeffrey Glenn group, we're working on a variety of different kinase inhibitors. And they didn't want me to show the structures on those projects. They're all in active lead optimization right now. But I can tell you that what we do is we do microsome incubations within the Jeffrey Glenn's group. And LCMS, we have a Thermo Fisher Orbitrap. And then the engineering group was sending their samples for incubation and LCMS to a CRO in China, Chem Partners. And then they sent me all the raw data. I put together the structures for them. And this just illustrates how I did this back on a project that we published some time ago from Roche. And what this early metabolite ID gives you is just a quick shot of the most common, the most likely metabolites that are formed quickly in the liver, and what the chemist might do as their next molecule to change the metabolic properties. And so that's really all I'm going to talk about on the early metabolite ID testing. That's probably mostly what Stanford's interested in. But I'm going to concentrate on this development issue, metabolites and safety testing, because it can generate some really interesting stories. So what's the deal with metabolites and safety testing? No one worried about this until the last 20 years. But metabolites can contribute to pharmacological activity. They can contribute to toxicity. They can contribute to drug-drug interactions. So regulations were promulgated starting in about 2002 with a white paper in toxicology and applied pharmacology. And more or less final guidelines have come out in the last couple of years from the FDA. And what are those guidelines? So this is the international guidance. Non-clinical characterization of a human metabolite is only warranted when that metabolite is observed at exposures greater than 10% of total drug-related exposure and at significantly greater levels in humans than the maximum exposure seen in the tox studies. So we're worried about things that weren't in the GLP tox studies that humans are going to be exposed to. Such studies should be conducted to support phase 3 clinical trials. And some metabolites won't be of concern. Most conjugates, you won't have to do any extra testings for glucuronide sulfate conjugates. And in general, these regulations are more or less ignored for oncology applications. So a decision tree that the project team can use to find out if this is going to be a problem for their drug. You measure the circulating metabolite at steady state, and then you have to do a quantitative experiment. Is it less than 10% of the total drug-related material, or is it greater than 10% of the total drug-related material? If it's less, you're not going to worry about that metabolite. If it's greater than 10% of the drug-related material, now you have to do a quantitative analysis in the toxicology species. So is it formed in any one of the tox species at all? If the answer is no, you're going to have to do toxicology testing on the metabolite. If the answer is yes, then you do a more accurate quantitation. If the exposure does not approach the human level, then you're going to have to do tox testing. But if the exposure in the animal approximates, and we've come to use the value of 0.5, half of the level that's in human, it would be an acceptable level in the tox species, then you don't have to do any additional work. So I'm going to illustrate this with a recent study that we just completed, and the article has been accepted to the premier journal in this field, Drug Metabolism and Disposition. And I'm going to spend some time telling you about the project team here. So I wrote the article and I completed this structure determination work. And I was brought into this by a metabolism scientist named Stephen Smith. Many of you may know Steve. He was my metabolism mentor at Roche, and he consults widely in the field. Mike Saporito and Gregory Busey were the scientists at this virtual pharmaceutical firm, Marinus, in Pennsylvania. And Mingbao Zhang was their chemist. He had his own little chemistry lab. Julie Wren is a principal at Quintara in Haywards, and she did the cold DMPK studies. Mike Fitzsimmons and Ping Yi are scientists at LabCorp, formerly Covance, the premier lab for doing carbon-14 work in drug metabolism. We had occasion to do an accelerator mass spectrometry experiment at Pharmaron, and so Stephen English is the principal who did that work. We had to use NMR to determine the structure of our metabolite, and the premier NMR lab is Novatia in Pennsylvania, and Adam Carter was the PhD scientist working there. And then finally, Tom Bailey, a big name in the drug metabolism world. He headed up drug metabolism at Merck for many years. He wrote the first draft of the MIST guidance, and we brought him in very specifically to hold the hand of the senior management through this whole nightmare issue. So what was the molecule that this company was working on? It's called ganaxolone, and the structure is here with a little asterisk showing the position that we carbon-14 labeled. And it's a simple methylated analog of the natural neurosteroid allopregnanolone, and it's in development for epilepsy in children. And it's not a new molecule. It was first presented by a California company, Consensus, in 1997, and people have been working on it ever since. Carbon-14 metabolism study was done in 2009, and it looked like things were pretty promising. These are radiochromatograms comparing mouse, rat, dog, and human liver microsome incubations. And the parent drug, see if I can work this out. There we go. Yeah, the parent drug comes out in this chromatogram toward the end. And the major metabolite, which was identified as M1, also seemed to be present it was a major human metabolite, also seemed to be present in the tox species. The scientists back then were able to identify the metabolite because comparable metabolism had happened in allopregnanolone. They were able to synthesize the standard. So M1 is a known structure. It's what we expected to be the major metabolite in this case. There was a second metabolite called M4, which was due to ketone reduction of the 20 keto group. And so we didn't expect a lot of problems when we started on this project. But when we did our first in-man studies, we compared human and animal plasma, work done at Covance in 2018. We found an unknown human unique metabolite that looked like it was due to a double oxidation. And so FDA said, you guys have to do a carbon-14 study. And so they started that in 2020. And they dosed 300 milligrams of drug, 150 microcuries to 8 volunteers, male volunteers. They collected excreta and blood out to 30 days. And you have to keep the patients in the clinic because you don't want the radioactivity going out into the community. And usually you want 90% recovery before you release the individuals. And they were not able to get that. They only got up to 70% recovery after 30 days. And most of it was in the feces, very little in the urine, but the total recovery of 70% means there's still quite a bit. But it was deemed impractical to keep those patients in, and the level of radioactivity they're excreting each day is so low as to be not really detectable, and so not a hazard. So they were released, and we started doing the studies. And so the first thing you do is look at the plasma after isolating it from the whole blood, and you look at the radioactivity in every time sample, and you analyze the drug by bioanalysis. And so this chart on the right shows that the plasma radioactivity is very persistent. There is something that lasts for a long time in these humans, and it's not the drug. By bioanalysis, the drug disappears very quickly, which we already knew. It's quite a metabolically unstable drug. That's why we have such high dose rates. And I just include down here, I don't know if you can see it, the dosing regimen off the internet. They dose day 1 to 7, 150 milligrams, and then 300 milligrams, then 400 milligrams, and then 600 milligrams to get levels of drug that will work in the indication.
They know it wasn't just stuck in the fat.
Very likely is stuck in the fat. It's like cholesterol. Yeah.
So it could still be the drug.
No, it would partition into the plasma. You would see it. It's completely undetectable after 4 hours by mass spectrometry, which is very, very sensitive. If you look at the numbers, where this is a log y-axis, there's very little drug compared to the moles of radioactivity at any time point. So we look at the plasma radio profiles, and then the way we do that is we make what's called an AUC pool. They take all of those individual time point plasma samples, pool them according to the total area under the curve. We did it first from 0 to 120 hours, so we'd have good signal for the detectors. And then we do LC and fraction collection with scintillation counting for the radioactivity, and a split flow into the mass spectrometer to identify the metabolites. And this is the curve that we got. And it shows one big metabolite, which was labeled M2 and was shown to have a co-eluting component M7, lots of other metabolites. And the project team was sort of hoping they could avoid the missed problems by going out to longer time points. And so we did one more experiment where we pooled from 0 to 720 hours, hoping the big peak would go away and it wouldn't exceed the 10% threshold, but that turned out to not be the case. When you go out to really long time points, the total amount of radioactivity in the samples gets really low. You have to use the extreme sensitivity of the accelerator mass spectrometry, same tool that's used for carbon-14 dating, to calculate measure the radioactivity in these fractions. But it works quite well. And you get out a chromatogram that you can compare to the chromatogram that you had before. And so we still had one or maybe two peaks that we would have to worry about as being over 10%. So we call this metabolite M2, and then let's go back and look and see if it's in our preclinical animal species. So we tested plasma from pediatric patients and from preclinical tox species. This was work done at Quintara in Hayward. These were all cold studies using standard mass spectrometers, and we used the exact same chromatography that they had used at LabCorp for the radioactivity work. So we could identify the M2 peak very easily, and we could see the M17 peak, which was the second peak in the radioactivity chromatograms. And now, working with a mass spectrometer, we have to show two chromatograms because the positive ion detectable metabolites are separately detected from the negative ion metabolites like stearyl sulfates. But then we looked at the animals and we confirmed the bad news. So in dog, mouse, male and female, and rat, male, the M2 was essentially undetectable. Female rat had a small amount of M2, about 1% of the level that's present in humans. So that confirms that we have a missed problem. M2 is a disproportionate human metabolite requiring toxicology evaluation. So if you're going to do toxicology, you have to have drug. We needed to work out a synthesis. Can't work out a synthesis if we don't have a structure. So this is really when I was brought in to help figure out the chemical structure of this M2. We knew from the mass spectrometer, first we thought it was a double oxidation. Later we were convinced ourselves it was a single oxidation, less two hydrogen atoms. It didn't form an oxime. That was a simple chemical experiment that we could do with the chromatography that we had. And so the ketone group at the 20 position seems to be reduced. There were two exchangeable hydrogens, an easy LCMS experiment to do with deuterium exchange. And we knew that the molecule eluted between M1 and glutathione, and GNX on the chromatography. So that told us something about its chemical properties. Unfortunately, it was not formed in human liver microsomes or hepatocytes. So none of the typical, usual in vitro or preclinical animal experiments that you would do in drug metabolism would have shown up this problem prior to first-in-man studies. And also, unfortunately, MS and MS of sterols is not really very useful. The steroid nucleus is very stable, and about all these things do in the mass spectrometer is lose water, multiple waters. Even when you put lots of energy into them in an MS experiment, all you get out is a statistical fragmentation of the backbone that doesn't teach you anything about where biotransformation spots might have occurred. So what can we, that's what we had hoped was going to happen with ganaxolone. But unfortunately, something about that one little methyl group changed everything. So where are the sulfates? And I had to say at this point, with my years of experience in drug metabolism, I couldn't predict whether the ganaxolone tertiary sulfate would even be stable. It wasn't, tertiary alcohols aren't a structure that medicinal chemists use very often. And so I didn't have any instinct for whether or not that would even form or be stable. So one of the advantages of being associated with Stanford is you've given me a SciFinder license. And so I can draw in a tertiary alcohol sulfate and see what's known in the literature. And I found some really interesting articles. First one I found was from an old friend of mine, Jack Hennion, who was one of the earliest pioneers in LCMS, and he specialized in looking at sterols in horse urine and people that were doping horses. And of course, that also leads into people that are doping humans. And so I found a series of what I call Berry bond compounds that have 17 tertiary alcohols where they've made testosterone analogs with a methyl group. And so those had some very interesting properties. When they were doing GCMS to see if people were doping themselves in that during the Olympics, they saw an epimerization, an odd peak in the mass spec that looks identical to the drug. And so they did the biochemistry to figure that out. And what they figured out is that it forms a sulfate at the tertiary center, as we were curious about, but that sulfate's not stable. It spontaneously solubilizes to the sort of carbocation that we all remember from organic chemistry that decomposes through methyl transfers and hydrogen losses and even recapture with water to give the epimerization. And so you get a mix of different things. And as soon as I saw that paper, I knew what was going on in ganaxolone. So next thing to do, call up the medicinal chemist that we had in the group, Mingbao Zhang. Mingbao, can you do this chemistry? And he said, sure. There was a literature procedure using pyridine and SO3 for sulfating a tertiary hydroxyl. And he quenched that reaction in water, sent the reaction mixture off to Julie at Quintara. She shot it on the LCMS, and we found a bunch of metabolites. We speculate that we could even see a little bit of the sulfate that hadn't decomposed. But the major things that you saw in that reaction mixture were unreacted ganaxolone, an epimer, which we labeled as M34, and a new metabolite formed by dehydration, which had a major and a minor isomer that we labeled M5 and M28. So that chemistry was clearly what was going on in our drug that doesn't happen in allopregnanolone. So we were able to synthesize M5. Mingbao got us a nice pure sample of it. We did human liver microsome and human hepatocyte incubations, and voila, there's our M2. So we now know that first you have to have this dehydration reaction take place, and then microsomes can convert the drug into the metabolite that we see circulating in humans. At this point, we decided to do an NMR structure. So we took C14 guided isolation. We had to use 200 grams of human feces, extract it with methanol, do prep LC 3 times. We isolated 105 micrograms. This was work done at Covance or at LabCorp, and they sent the 105 micrograms to Novatia, and they compared the NMR to the NMR of the purified M5 to identify the position of the double bond in the A ring, and then to show that the source of the oxygens was an oxidation of the now allylically activated methyl group in the A ring. And I won't go into the details of how clean and clear that NMR work is, but it was really very elegant. So now that we've got the the structure, we can do the synthesis. And so Ming Bao did this, and I'll spend a little bit of time going through it because my other chemistry mentor was Carl Gerasi, and of course he was a sterol guy, and he got the birth control pill going at Syntex, and maybe that's the reason was a chemical industry in Palo Alto for a number of years. And so I'm interested in sterols, and the functional group change chemistry hasn't changed that much from the stuff that Carl worked on 50, 60 years ago. So you start with pregnenolone or pregnanolone that's reduced in the 4,5 position. You make it epoxide, you open it, you get an iodo and then an acetate hydrolyzed to the, which is pretty easy to get to the acid. And those steps all went very well, and they gave us access to at least one other metabolite, M42, which we had observed in the chromatograms. And when Ming Bao went to do the reduction of that ketone group, he always got the wrong isomer doing it chemically with borohydride and other reducing agents. He just couldn't get the right isomer. We were able to isolate the right isomer by PrepLC, but for scale up for the toxicology studies, we had to hire another company, another CRO that's called HIFA. They're in the UK, and their specialty is synthesizing drug metabolites. So the big pharmaceutical firm that finds a problem like this, they can go to HIFA. The HIFA will screen all different cytochrome P450s, including bacterial ones, and come up with a way to scale up and get the synthesis of a metabolite. And they know everything there is to know in this field. They know how to use this alpha keto reductase 1A or 1C, which is really a reagent of commerce that's used widely for ketone reduction in the synthesis of drugs and metabolites. So they worked on that chemistry. We got a good synthesis of M2. Ming Bao was then able to convert that to the steroid, which was proven to, or the sulfate at the 20 position, which was proven to be our other major human metabolite, also disproportionate, M17. And so what's up with the safety evaluation of M2? So this is the latest information from Marinus. They had to make 6 kilograms. Had to do an in vitro AIMS, a chromosomal aberration, and in vivo micronucleus. They've completed 1 month and 3 month tox. I think 6 month is still in process. They're doing a 6-month CARC study. Any of you that know toxicology, you know how easily it would have been for something to kill our drug right there. So this isn't something you really like to have to do, but it's worked out so far pretty well.
Is one species?
I think they only have to do one species, yes. I think they're doing rat. Luckily, there was no functional activity at the GABA A receptor. So that 3-hydroxyl group is critical for the activity of the drug. And so there's no functional activity in this metabolite. And it has greater than 99.9% plasma protein binding. And that explains its slow clearance. Of course, it's also bound in tissues. It's in the fat, like cholesterol. But it's just doesn't get excreted very quickly compared to the other sterols. The parent drug itself, the M1 metabolite, is excreted as a glucuronide into feces and doesn't show up in the circulation at all. So then a validated bioanalysis confirmed that the M2 in the rat is about 1% of the M2 in the human. And so far, the toxicology studies, it's been well tolerated with very high bioavailability in rat at 0.30 and 100 milligrams per kilogram. So far, so good. Just to complete the story, there was some concern at the FDA that neurosterol sulfates can cross membranes and be active, in particular at the GABA A receptor. So they challenged us on this sulfate, but because it's a sulfate of a molecule that has no activity at the receptor, so far they have agreed that's not a molecule we need to do any toxicology testing on. Thank you. There were two other dehydrated metabolites, M5 and M6, that passed the 10% threshold in the early 1 to 120 hour studies, but they disappear in the long-term study. So far, we don't have to do any additional studies on those two metabolites. So with all this information in hand, we can lay out the biotransformation of ganoxolone that we couldn't do before we did this synthetic work. So the molecule is cleared by cytochrome P450-3A4, and we knew about that. That's the one we knew about as we were developing this drug. We'd done drug-drug interaction studies with that enzyme, and so that was all in place for the drug, but we didn't know anything about these other clearance pathways. And so the keto reduction does not seem to be a major pathway for the intact drug itself. It seems to happen maybe after the dehydration. We're not absolutely sure. But the thing that does seem to be very important is the sulfotransferase reaction. And we haven't done the specification of which isoform of the many, many different human sulfotransferases there are. But we speculate that the reason the animals don't have this metabolite is that specificity of that sulfotransferase. But once we've got this information, now we can put the whole biotransformation package together. We can identify the metabolite M5 and M6, M2 and the M7, some of the other minor ones, the M1, which we knew about from the very beginning, and these other peaks we see in the chromatography. We can explain all of them and do a complete drug metabolism analysis. So, conclusions and lessons learned. This was kind of a nightmare for the company. You know, they're a little virtual company. They're dependent on consultants, and the company that was doing the carbon-14 work doesn't have the capability of doing the intense structure determination. So they needed, you know, Steve Smith and me and Tom Bailey to put the structures and to do this whole project for them. And they could have lost their drug right at the time when they were filing their NDA. Now, in Big Pharma, when I worked with Genentech, when we were putting together our strategy at Roche in 2008, before they shut the site down, we were talking about how we would discover these issues in phase 1, right? You would get the samples from the first in-man studies, and you would do the LCMS profiling. You would see that M2. You would find out it's not in vitro, it's not in the tox species. And, you know, you could consider at that point, maybe we need to consider another analog, or at least work out the synthesis and start doing some tox studies, see if we've got a problem. But Marinus didn't do that. You know, they were on bare bones, do whatever they had to do to get their molecule moved forward. Secondly, definitive drug metabolism projects require a big team of scientists. And at a big pharma, they have a big team of scientists. I presented this seminar to my friend Cyrus Kojaste at Genentech, and he brought in all the people in his group team, and he's got about 15 people that do these kind of studies. If you're a virtual company, you're going to be relying on CROs, and those CROs need to be orchestrated, and that's what I did in this project. And so I want to conclude by saying DMPK CRO management is a critical part of drug discovery. And I got one more slide that I'll use to introduce Mike, because this is something we worked on together 10 years ago. Little molecule that was discovered here at Stanford, RRX-001 in the radiology department. And they were developing it as an anti-cancer drug or as a radiosensitizer. And it's still in development, but the metabolism was very complicated. it alkylates every thiol that gets its hands on, including hemoglobin. And so in our paper that we published in 2012 in Drug Metabolism and Disposition, we did the proteomics to show that it specifically alkylates cysteine 93 in the beta chain of hemoglobin. And we had done the work to identify and synthesize the glutathione addict of this drug that was used to monitor its bioanalysis, because you cannot detect the drug in any animal species that reacts so quickly with thiols. But that was a fun project, and it's still ongoing. And then my last slide is a paper I published with the people at Genentech, where we were doing a physical organic chemistry study on what we could do to predict LC retention times and use the retention time in HPLC to help identify metabolites. And it's really useful in cases like ganoxolone, where you don't learn from the mass spectrometer, where you've got a number of possible isomers. And in the case of a lot of small molecules, you can get an aliphatic monooxidation, or an aromatic monooxidation, or a nitrogen monooxidation. And you can see all of those. And very often a person experienced can just identify them from their retention times, because those biotransformations affect their hydrophobicities in very predictable ways. So anyway, I'll stop there and ask if there's any questions.
Well, and I came late, so maybe what I'm going to ask has already been covered. But I do remember that, throughout the different times, different metabolic studies throughout the ages, all drugs often don't have this kind of support in the file approved FDA. Do you have any advice for when projects repurpose a drug, different route of administration, and often the metabolites studies are completely...
I haven't asked that question of the agency, but I think the answer would be you're going to have to deal with this for a new indication. I mean, you've got years of tox studies in man by now. Maybe that precludes worrying about this.
Well,
I bet it's going to be kind of case by case, unmet medical need. If it's cancer, we aren't going to worry about it.
Right. But it's definitely an issue. So
virology, I've heard different stories on whether or not MIST is not a big, huge issue with virology, where you're only going to take a drug for a few days. But if it's an HIV medication and you're going to take it for the rest of your life, I bet they're going to want to.
An issue. So it really doesn't matter whether.
Because you're right,
if it's more than 10%. So when you repurpose you may have different metabolites.
Yeah.
So that's something. Frankly, I highly encourage think about metabolites even with drugs that you think there's a lot of information, because the reality is metabolite studies are not available.
And something you're repurposing, it should be very easy to get the data if you can get clinical samples and look at what's in there.
Or, we use that term a little loosely, repurposing, right?
Yeah.
Because we really, depends, right? Is it the same route, right? The route changes, the metabolism can change. I mean, we've all over the years seen instances of this. I mean, it's not necessarily common, but it occurs, So I think, do you change the route? Do you change the duration? The disease indications. Those latter parts aren't necessarily to do with the metabolism, but if the route changes, for sure. And in general, what I've seen is a request to fill in the gaps. You know what I mean? I mean, if something was approved, whatever, 30 some odd years ago, it may not have been approved with the current level of scientific rigor that we employ. So bring it up to a standard. So I'd be prepared for questions. That's the point, really. I wouldn't assume, I guess, that it's a slam dunk, unless it is a slam dunk. You're changing an excipient, you're still giving it orally, the kinetics don't change. You have those data, right? You have those data. But as changes arise, there are gaps, and as I said, what I've had in terms of feedback from the agency is to be fill in the gaps, fill in, make sure we understand.
That's a role.
The soccer ball.
Right. So in impurity analysis, it makes a difference what the dose is going to be for the drug substance. If it's being given at 100 milliliters, I forget what the threshold is. There's a different level of impurity that's required for full characterization than if it's a highly potent drug. Yeah. So my question is it 10%? This 10% threshold, is that true regardless of what? As
far as I know, it is. There are, in the reactive metabolite business, we always talk about 10 milligram drugs. You don't need to worry about reactive metabolites. But I don't think the regulations address dose with MIST. But it's certainly something you might bring up and say,
Okay. All right. My other question is a chemical one. The acidity of that carbon is leading to the elimination of the sulfate and forming the disulfide bond. That's not very acidic. You don't see any of the other disulfide. Not disulfide. The olefinic bond that can form from the elimination of the sulfate.
We saw two isomers, two main isomers. I think there's minor isomers too. I think I had all the same products of that solvolysis that they had seen in the literature. I just concentrated on the major ones that explained it.
All right. Okay. Thank you.
To what extent is now pharmacogenetics coming into play in these kinds of studies where you have the possibility of polymorphisms and so on affecting drug metabolism?
Well, we had to worry about that because we're using 3A4. It was one of our, actually, we thought it was our only clearance mechanism. So they had to do a lot of DDI studies in that case. I don't think anybody has ever had a DDI study on a sulfotransferase based. It's not usually a major clearance pathway, but I guess it could be. But I don't think that would be something that people would jump on. Sulfates aren't usually toxic.
So
drug interaction studies are kind of, each case is different.
I was thinking more in terms of the phase 1 clinical studies where you want to get the drug metabolism. If you don't have kind of a good spectrum of population, you may be missing a particular high metabolizer. So to what extent is that
it really is not addressed. Carbon-14 human studies have always been, white male volunteers. And like a lot of toxicology studies, right? You want to compare data to the historical data. And the DDI questions are dealt with in different forums.
And that was just to add to that, at the very least, right? We do this in vitro. Just for a second, we do this in vitro.
Yeah.
So we have an appreciation of the various, at least for SIPs, right? SIPs and a little more. We have an appreciation for what the metabolism may involve. So that's the beginnings of the appreciation because it's understood, right, of that variability. I'm not a great girl.
Thank you. I was going to kind of come back to the first point, tox program that presumably FDA asked for. And I was wondering if that was because it's a pediatric indication. And, I do think it will matter what the population is, what the indication is, how long you'll be taking the drug and things like that. Yeah. And with pediatrics, everybody's a little bit more lot
Yeah.
more cautious for good reason.
I was pretty isolated from the whole regulatory discussion, so I'm sure you understand what was going on between Marinus and FDA better than I do.
You mentioned in animals that the M2 was higher in the female mice.
Mhm.
Would there be any concern that there might be a different metabolism in women versus men? And would they consider using women?
I don't know. I think I might let Mike answer that one again. These are complicated drug development problems. The toxicology studies tend to be done very strictly. And, you know, this is going to be a drug for children mostly. Our differences in metabolism, I mean, we haven't done metabolism studies in children. That would sort of be considered unethical, I think. They are the patient. There were patients. This drug is being used because it's an unmet medical need. Yeah, we had no trouble getting lots of clinical samples.
But the trials, the human trials, right?
Yeah. Oh, yeah, they would children. And they probably are measuring the differences in the clearance of the drug in male and female. I mean, that would be the experiment that they'd be doing.
So what was the cost of all this?
Well, you could guess 6 kilograms. The toxicology studies,
The whole thing.
I have no idea. I'm totally isolated from the business end. I'm just a consultant, just 5,000 miles away from the company.
I guess up to $10 million. Yes, for sure. And we never used to do this stuff before. We had HPLC and high density mass spec, and somehow all those drugs are still around. Isn't that amazing?
Just like earthquake safety in your house. Yeah, a lot of things happen with civilization.
I know who to call if I ever wind up in this problem, though,
Yeah.
with this.
Yeah.
One of the things that I was thinking was in the rat, it was like 1% or something, this metabolite.
Yeah.
When you are doing the PK studies, single dose PK studies, or like pilot talks, whatever, I would think that would be the stage where you could get the samples and do the analysis.
Absolutely.
Right?
Yes.
GLP talks you cannot, because there are so much restrictions and it is really hard to get the samples and do all these analysis.
That's what they would be doing in big pharma. They would be getting the early PK studies, and they wouldn't have found this metabolite because it's such a small percentage of the rat. They would have ignored a 1% metabolite. But the first in human studies, they would have found this.
Exactly. Yeah.
Phase 1. They wouldn't wait till phase 3, and they've already spent whatever.
So one of the big concerns with small biotech companies is the cost of doing the bioanalytical work.
Mm-hmm.
So the method needs to be at least qualified to the level where the program is, and also the cost associated with the other things. So maybe that is something that if there is a price breaks for small biotech companies that could be affordable for them to do this at the early, then you don't need to go through a lot of other things. Just wanted to make that statement.
Don't know how the FDA feels about that. It doesn't sound like an argument they like.
No.
A very patient person back here.
Okay.
I was just wondering if there are any examples that this is like kill the project where maybe in phase 1 there didn't seem to be.
Mike?
I want to say yes, but it's always a multifaceted discussion. What's the need?
Yeah.
The citizens, but how they the tribulation that they have to go through. there certainly are examples of human metabolites that don't show up in animals. So it's not necessarily common, but it's not remarkably uncommon. And histamine that showed up in Japan, the metabolite factor was the signature problem, right?
Yeah, I guess.
I mean, the worst case is when that is either active or potentially toxic, right?
Not at the yeah. I mean, maybe a more common case is the reactive metabolite idiosyncratic tox kind of problem, where it doesn't show up in animals because you're only dosing 100 animals. It only shows up when you give it to 1 million humans. All right, your turn, Mike. Oh.
I wonder if they had looked at non-human primates early on in the in vitro screening using hepatocytes or something, if they would have picked this up, because it might have. I don't know. You didn't mention anything about non-human primates, but
we did monkeys. It wasn't formed.
Wasn't? Okay.
Yeah.
That answers that one then.
Pigs had a little bit.
All right, so our next speaker is Dr. Michael Taylor, and Mike has been a very long time SPARK advisor as well, and has been just a tremendous asset and has helped so many projects get over the bumps along the way. So Mike is the founder and principal of Nonclinical Safety Assessment, which is a consulting firm that focuses on safety evaluation of drugs. He has more than 30 years of biopharma experience, including career starting at Syntex Research, and subsequently held leadership positions at Roche Bioscience, Direct Corporation, Protein Design Labs, Alexa Pharmaceuticals, Limerick Biopharma. Is currently vice president of toxicology at Alumis. And as I said before, has been a tremendous help here at SPARK. He got his PhD and master's degree in toxicology from Utah State, did his postdoctoral training at CNRS in France, and also the National Institute of Environmental Health Sciences, active member of the Society of Toxicology and American College of Toxicology, and like Bill, he has a gazillion publications. So here we go.
Hey, thanks.
I'm waiting for the light to turn green. Is it working?
Oh.
It was working. This is the one that Bill was using. Try this one also.
Okay, there we go. Okay. Well, thank you very much. It's a pleasure being here tonight. I always enjoy being here and having the opportunity to talk once in a while. And so for tonight, really, I mean, maybe a little bit in the vein that Bill was talking to and continuing that in terms of activities that we have in place to support drug development in the United States and around the world. And so specifically tonight, talking about non-clinical safety evaluation and drug development. And I mean, the overall focus tonight really will be toxicology evaluations for small molecules. But this is basically forms a paradigm for testing of would-be pharmaceutical products, you know, whether they're small molecules, whether they're large molecules, whether they're devices for delivering drugs. The concepts, I mean, are applicable broadly, and talking to this from the point of view of small molecules, probably the most one has to do in this arena. Toxicology testing has to do with small molecules. I mean, some of you that appreciate the other pharmaceutical modalities, for instance, going like to the other end of the spectrum, or cellular modalities I mean, they're not really such that the standard traditional toxicology paradigms we have in place for testing work. So but the concepts of starting with, we have to do something, right, to de-risk the molecules, the modalities that are introduced to humans. We have to do some level of de-risking. That paradigm can be based on the learnings and what is required now for small molecules. So toxicology, you know, it's a discipline, really. I mean, when I started, it wasn't really available in undergraduate. I hate doing this because then it's obvious how old I am. It wasn't really a discipline that was available at the undergraduate level, and actually, save for a few medical colleges around the globe, maybe more in the United States than really around the globe. It was a multi-departmental activity usually within medical schools, you know, that had good pharmacology training, because really, in many ways, toxicology is the continuation of looking at pharmacology, the exaggerated pharmacology, the suprapharmacology. But nowadays, the trainings are open at the undergraduate level. The utilization of trained toxicologists in a variety of areas, from food to agricultural, various industrial applications, chemical industry. There's broad use of toxicologists. So, you know, I think probably when drugs are developed, a very common term, optimization. We're optimizing everything. We're always optimizing, right? And the toxicology activities, they fit into that scheme of optimization, together with the works that folks like Bill do, and the chemists. We're looking for that sweet spot. It's not perfect all the time, but it's a balance of trade-offs and in relationship to what we're actually trying to accomplish in terms of disease treatment and prevention. And so, yeah, that's the piece I sit in usually. Through my career, I've been fortunate to usually begin with the folks that start this. So the medicinal chemists, I'm very often in discussion with. Folks like Bill, very often in discussion with, and to the extent I can contribute as a toxicologist, I do, you know, and we begin to do evaluations these days, evaluate the totality of data these days, you know, much more than we did as I started in my career. So what's the purpose? You know, a lot of what I have on this slide is the early purpose of doing good toxicology evaluations. And one, and really maybe right at the top of the list, is to eliminate those molecules that aren't going to make it. You know, it's a crapshoot, our industry, really. It's a very difficult one. You know, millions of people have made millions of predictions, and the bulk of them fail. You know, the molecules fall out. I mean, it's amazing how big the garbage can is for molecules that failed. Figure out whether or not the molecule is going to work, have a too large of a safety liability, the better. But the early tox evaluations, in part in addressing that first point, they're identifying potential target organs and systems, begin to put the thinking in place, how much more I may have to add to these types of evaluations. And then ultimately, what might I have to do now in the clinic, right? I mean, because everything I do from this point forward has a cost, right? So sometimes these costs are prohibitive, really. Identifying an extension of that, identifying potential monitorable parameters for clinic. Sometimes these are understood parameters. Invent isn't the perfect word, but we'll create new biomarkers, right? It's that opportunity for such. Most importantly, the first tox studies that are done to support clinical entry set the dose. They set the entry dose for the very first patients that are dosed. So it's good to and I'll talk later, but to have well-designed tox studies in order to enable a reasonable selection for a starting dose. You know, you get that wrong, it's not always obvious in the beginning. You get that starting dose wrong, and it may take you a lot of subjects and a lot of money to get to that sweet spot in the clinic. So being as aggressive as you can in the starting dose and doing that safely, you know, there's an economic driver there as well. The tox studies, they actually, in addition to setting a starting dose, they tell you whether or not you might wind up with a hard stop in terms of a limit on the clinical exposure. Things that are non-monitorable or have steep dose response curves in terms of the safety liability. They often require a setting of as I said, a hard stop. Thou shalt not dose the subject beyond such and such a level. And usually we're talking about a 10x margin in that regard. So you begin to appreciate, do I have enough range here, enough latitude, enough freedom to operate when I get to the clinic? Help with identifying at-risk populations, carcinogenic potential. Ultimately, for long-term use molecules, we're hoping to appreciate. And then ultimately, for those of you that appreciate the labels, right, that go on to drugs, there's always some information relative to that last point, carcinogenic potential or reproductive liabilities, for instance, and sometimes even beyond that, depending upon the nature of the tox liabilities that are known or uncovered. And maybe, again, just to reiterate, things that actually wind up with no-go decisions, genetic toxicology. Though it's interesting, I'm just working, first time in my career, I'm working with something that is potentially genotoxic, but it's a single use, and it's for a diagnostic tool that's if it works, it's remarkable, right? It'll change the way in a particular area how medicine is working. But usually, we don't advance molecules that are positive in the sense of being mutagenic. Anyway, we don't advance them. Inadequate therapeutic indexes outside of oncology. Now I'm talking primarily to molecules that are developed for chronic non-life-threatening indications. Inadequate therapeutic index. Too much risk in terms of the toxicity to vital organs. This can certainly be a showstopper. I spoke to this initially, but non-monitorable toxicities with therapeutic indices, so the separation of the tox liability and efficacy of less than 10 can often be problematic. Lastly, idiosyncratic, not well understood or behaved toxicology is often difficult. Crazy things in one species, not in another. A lack of clear relationships, these types of things. Again, just to reiterate, I mean, the bulk of what I do, even though there's been quite a bit of discussion, certainly in this last year, around the use of animals in pharma research and certainly toxicology. There are a number of rules and groups that we work under with very high scrutiny. I mean, I always thought the kid that the animals get better care than a lot of people I know really these days. So, but just the awareness, when we're using animals, I mean, basically should have an appreciation for the use of those animals and make sure that they're being well taken care of. Just again, maybe just talking to animal use in drug development. Again, from where I sit in my experience base, and there are papers like this is not in isolation. There is a reasonable concordance, between animal testing and the prediction of human toxicities for pharma. I mean, it approaches. The next slide actually talks a little bit to that. But collectively, when two species are used, a rodent and a non-rodent, it's in the vicinity of 70%. And, this is appreciating, this is a very large mass of data that went into these types of appreciations. Are they perfect? No. But there are many cases in the past history before we really were scrutinizing these things at the level that we do today, where mishaps occurred clinically, and they still do, actually. It's not an absolute rarity. So the use of animals has been beneficial. Some of you are appreciating the law passed this year that said the FDA does not have to require animal testing. Well, from where I sit, they never had to require animal testing. I mean, I have a case of a monoclonal antibody that was approved without really any toxicology testing. Animals were used, but no toxicology testing. And the FDA, I mean, we have a general paradigm we work under, but the FDA can tell you not to do something or to do more. It's always really been in their purview. Just to be clear, I mean, the distinction between the early safety evaluations and the later time points. In the beginning, you don't really appreciate efficacy. I mean, you understand it from the dish, you understand it from animal models, but you don't in people. I mean, that's obvious, right? And so in the beginning, it's about manageable risk. We do the tox work, we kind of set some parameters in place, some ranges of doses that we think are safe. And we start, and then over time, over the next years, we weigh what we see in terms of safety liabilities against what we see in terms of efficacy, and we always come up with a risk-benefit appreciation in that sense. And then all of that is dependent on what the disease is. Some diseases, oncology, and historically greater tolerance for tox liabilities. Urinary incontinence, the other end of the spectrum, much less tolerance for safety issues to be a common component of the therapy. So again, in the end, it's always a balance, really. So a lot of you here, good laboratory, we do the tox work. It's all done under GLPs. What are GLPs? GLPs, I don't know how all of your labs work. My major professor made us work under GLPs from the beginning. I mean, basically it was good practice. I mean, for doing rigorous work, I mean, reproducible work. We've all heard that criticism in the last year. Your work's not reproducible. So working under GLPs, for me, it's been a standard. It's not a high dollar thing that adds to cost of a 3-month primate study. I mean, this is just it's bells and whistle checks. It has to be done. Studies need to be conducted under GLP regulations. Certainly when they're pivotal toxicology studies. It's a rare day that we're basing decisions on non-GLP tox studies. It happens, but it's a rare day. So studies are done by according to GLPs. I spoke to it initially when we're moving forward with potential drug candidates. We try to introduce some tox screening early on. There's a short list of things that can be done. This list is really driven by the appreciation of what we're dealing with, right? I mean, the scientists that bring the molecule forward are in the best position. What could be a tox liability? I'm working on a drug class right now. I mean, potentially hematologic liabilities, toxicities were a known component. So let's do some screening, right? The companies that can do that type of screening are there, so add it to the list. So you begin to think about what are potential tox liabilities early on. Ultimately in developing a chronic therapy, it's a long list. I should say it's an expensive list. It's maybe not that long a list, really. There are a lot longer lists. But these days, and I have actually a slide here for cost to approval, approximate cost to approval when you're using primates these days. To me, you'll see it in a little while. It's a bit staggering because I think it's more money than it cost when I started to develop a drug to registration. Again, dating myself, But there are a number of study types that are done. I mean, chronic daily therapies. I mean, the expectation is to conduct tox evaluations up through, actually up through 2 years. I mean, really, when we talk about the carcinogenicity studies at the bottom of this list. But we do repeat dose tox for, many months. There are a battery of safety pharmacology studies. This battery used to be quite a bit longer. When I started, we called it secondary pharmacology. We almost looked at every organ system in the functional system in the body as part of development. But these days we focus on just the brain, the lung, and the heart. Genetic toxicity testing, phototoxicity, depending upon the population that you're ultimately going into, maybe juvenile animal work, reproductive tox studies, particularly when women of childbearing. Is that rain or is it just wind? We can't hear the rain in here, right? Oh, from the middle, right over there. Okay. The reproductive toxicity studies, and I'll talk a little bit more about those as we go. So I always tell people, Bill alluded to these types of guidances from the International Conference on Harmonization. But this, in my realm, this is the one guidance document that you read. I mean, this is basically lays the map out, what the expectations are from developing products from single use to repeated chronic use. And by way of just showing you the table of contents, you can get an appreciation of that. So this is the guidance, whether you're talking about small molecules or large molecules, proteins, monoclonal antibodies, not cellular products necessarily, but the large and small molecules. It's the guidance to first take a peek at. And in general, I mean, to develop a chronic therapy, and this is for the US and around the world, we're basically doing equal time in animals, or our time in animals supports same time of dosing in humans, So this is basically just a table showing that the expectations of studies, durations, the general tox work now, the study durations to support clinical use, the durations. Sometimes even today I hear, from people, they and that's that underscore in that first footnote to this table, that you can go beyond those limits that are set in that first table. That's a rare day. I mean, in my career, that was in the beginning. As I started, we were often able to go in parallel, so you could have an animal study ongoing at the time of a clinical trial, and only come back and report the data to the FDA, for instance, as necessary, right? So some untoward event occurs in animals, we have a safety report to the FDA. But nowadays, even when it says for serious or life-threatening indications, it's really more that case-by-case basis, really, for being able to continue dosing in people beyond the limits set in animals, outside of oncology. Again, just the expectations that are required to at the time of approval. And there's a little bit of variability, and these guidelines, the ICH guidances, are generally applicable around the globe. So anyway, you've got the molecule. You're ready to start your program. It's funny. I put this list up here of how you can figure out what you want to do, but really, it's the last point. That's the most important one. You talk to somebody who knows what they're doing. Just like when you're getting ready to make a molecule, you don't talk to the hairdresser. You talk to the chemist. I mean, that's how you make the molecule, right? So what I've talked to people that are experienced, in this day and age, there's tremendous depth of experience in all the areas, right, that we have for drug development. But for me, I guess very often what I look to, if I can find a similar modality and use molecule in the disease indication, so I look at the FDA summary bases of approval. I mean, they published this, and you can actually look what was done in the, for the clinical development trials. I mean, you can look to see what type of animal testing was done and get a sense of expectations. The only caveat I add to that is very often, particularly if the drug was developed by a large pharma company, they very often go beyond what the FDA actually required. They don't ask the FDA. They don't. They're more apt just to do what they think is best. So you just, if you, in reading the summary bases of approval that the FDA provides, you can actually wind up making decisions that go beyond what the expectations of the FDA are. So just like a warning. And again, I've touched on this as I've been talking. Really, the types of studies, what will be required is driven by what the therapeutic indication is. And the extremes are, right, for non-life-threatening chronic diseases versus life-threatening oncologic diseases, right? There's really quite a difference here. I mean, the need to get a treatment out there for a non-life-threatening, no real sense of urgency. There's no sense of urgency. So people, you do it all. You don't get a shortcut, right? You're working in a disease area that's life-threatening. I mean, really, right? The life expectancy is years. I mean, a rare pediatric life-threatening disease. The willingness and the wantingness from the regulatory authorities is high, right? So the guidances that are there demonstrate this when you review what's necessary for developing an oncology molecule in terms of tox versus a chronic non-life-threatening molecule. The third point in general, we conduct these studies in two species, a rodent and a non-rodent, and always there depends, particularly if we're talking about antibodies. I mean, it's always a good example. Oftentimes the target affinities are such that either there's a lack of homology or the species doesn't reproduce the pharmacologic effect, so the need for two species can be minimized or eliminated. And in general, these species that I'm listing here are the models that we use these days. Some of you may appreciate, I mean, these last couple of years have been pretty trying. The Department of Justice actually closed down one of the large suppliers of beagle dogs. And as a result of that, the company that owned the facility just got rid of it. I mean, they stopped, right? So the supply of dogs in the United States, around the world, was minimized drastically. And with quite a consequence, the biomedical research really in terms of timelines, costs. For me, for the folks I work with, I use only one supplier of dogs. I've only used one supplier my entire career. That's Marshall Bio Resources, upstate New York. They have probably the best beagle available for a variety of reasons. But again, as a consequence of what occurred, the resource was restricted. And some of you may have heard on the primate side, some folks from one of the bigger suppliers in China were arrested. I mean, when they got off a plane at JFK for, I guess the charges were fraud, ultimately fraud, that they were selling purpose-bred animals but they were actually wild caught. At least these are the charges, and it immediately, threw chaos into the availability of primates around the world. Even now, I use a lab down in Indiana. It's becoming a larger CRO, Innovative. But talking to Phil Downing, the GM down there, they're actually having to do now genetic testing on the monkeys to verify that. I'm not quite sure how this is done, but the genetic testing to verify that they're purpose-bred rather than wild caught. They're trying to figure out the breadth of assays to perform, so it'll be more than just one, to confirm whether they're purpose bred or wild caught, including they're measuring various trace mineral levels to substantiate they were on a particular diet, right? You get it in a purpose-bred facility versus wild. And I don't know, Phil told me something else. But all of this, right? All of this impacts the cost of animals and impacts the availability. And you'd be shocked. I mean, some of the CROs these days for primates are charging upwards of $60,000 per animal. I mean, it's a remarkable charge. I mean, so, you know, I hope as you go out and talk to VCs, you better be prepared. You need a lot of money, particularly if you're gonna be working with primates these days. So in general, what I've got on this slide is showing what's required to do a first-in-human study, around the globe, a first-in-human study for molecules that are intended to be given repeatedly, right? I should say, I mean, in the United States, you can actually, it's time for time. So if you have a single-use, you want to do a single-dose clinical trial, you can do a single-dose animal study and support that trial. There's a lot of risk in that, I'll tell you right now. And maybe just quickly, toxicology guys, people, they don't often do these single-dose studies anymore. So they're not actually always familiar with what might happen, and what might happen might be transient. And transient often doesn't really translate ultimately to tox that I worry about, but you don't know when you do a single-dose study. And if you're going to develop the compound for multiple dose applications, right? I have it in a slide later. I always call these early stages nonlinear. It's remarkable how profound the toxicology can be over the first 4 weeks of treatment. Every day counts. So you get other than doing a single dose bioavailability PK study, it certainly isn't going to provide a heck of a lot of ultimately of safety data from either the animal side or the human side. So in general, 2 to 4 week studies, safety farm studies that I discussed. Sometimes these are very often these studies can be brought into the general tox studies and kind of limit the need for them as standalone studies. Some genetic toxicity assessments, at least in vitro, and a phototoxicity assessment. And in general, that's what we do. I mean, there are always exceptions, and depending upon exactly what you're doing. So, it continues from there, as I said, depending upon the duration of usage. You know, ultimately, we're doing a 6-month rodent study and a 9-month non-rodent study. Again, if particularly when women of childbearing potential ages, right, are brought into trials. And I just learned today that a company I'm working with, a woman was involved in the trial and 65 and became pregnant during the trial. So when I think you have to think broadly these days, when women of childbearing potential goes beyond what we might have typically thought, right? But so the need for reproductive studies, carcinogenicity studies, basically still a requirement, but I'll talk in a couple slides later. There's a little more openness. I don't know if it's precipitated from the level of animal rights activity in the last couple of years or not. That there has been a similar movement in Europe that would taking, I think, a little more traction. But the FDA the ICH has actually issued an addendum to their carcinogenicity guideline to open the door a little broader to obtaining a waiver for doing such studies, such as a 2-year rat carcinogenicity study. And I have actually just been submitted the first waiver I've ever been involved with for a molecule. I'm waiting to see what they say. So what I thought I'd do maybe become a little more granular with how to put together good dose range finding studies. So the initial studies that we generally do to support ultimately dose selections for longer and longer term studies. You know, these are the least expensive studies to run, but on my book, they're always the most important studies because it's the first brick. I mean, this sets the stage. I mean, hopefully you're going to put a well-designed study together to appreciate the dose response range. You're going to appreciate the potential tox liabilities. You won't see them all in a 2-week study, but they certainly open the door. So the basic elements of the design are on this slide. I mean, you'll hear things about validated assays, etc. I mean, these are non-GLP studies. The assays that Bill talked to, they don't need to be validated at this stage. But we should be collecting the data, the initial data in terms of metabolites, as shown here. Sometimes I was just going to share real quick. Sometimes I hear people abbreviate these studies, like in terms of that toward the end, the anatomic pathology evaluations, to save money, right? Save money. It's a mistake. I mean, really, you look at 10 tissues, you miss 30. I mean, so you don't really have any. This is your first big survey study, unless you really are taking a good survey, you have the potential to miss things. So then you go to the next step, right? You spend more money, and you really could have stopped right here. So I always recommend that people basically look at the same set of tissues, the numbers of tissues that we do ultimately in the GLP work. Yeah, just a little more, just talking to the species. Again, the choice of species, is three things, right? We talk in terms of they should at least one, both ideally, but they should capture the pharmacology that we're going after. So you want them to reproduce the pharmacology. You want them to have, as Bill talked to early, metabolically, you want them to behave similarly to what the expectation is in human. And I'd say those are additionally, you want to pick a species where there's a very good understanding of background changes. This is particularly true when you get the pathologist is involved. When you stay with animals that we know, there's a remarkably large historical database, for instance, for a Charles River, for Sprague Dawley rats, right? Remarkably large for CD1 mice, beagle dogs, this particular cynomolgus, this particular macaque, tremendous database. You go outside those limits, you ask for problems. People get sometimes one animal has a finding, and it counts. You can't dismiss it. When you have a large historical database to draw on, you can appreciate. Maybe in that database, right, you have an animal. You encounter this lesion, at some frequency. So sticking with models that are known in the known space is the ideal. You can't always do it. Typically in terms of sizes, like these numbers are fairly standard. I mean, I find there's not really a clear specification from a regulatory point of view on, save a couple examples of how the end, what the end should be for tox work. But there are examples, but not across the board and across all countries. But typically, 5 rodents per sex I use, and 1 per sex for the non-rodents. Sounds like a small number, but it's 2. And I'll tell you, I've done that for 30 years. I mean, it's always worked. I've never had a mishap but it takes a lot of thought, right? In terms of dose selection, for instance, which we can talk a little bit more to that. Again, a question I hear a lot of times around the API. Does the API have to be GMP for toxicology work? And the answer is no. But if you make it GMP, it's real easy. You don't have to worry about it, right? I mean, so when I say non-GMP, it almost goes through the same level of rigor that in terms of characterization that it would if it were GMP. I just save a few. There's just a few things that don't have to be done. Methods that they're used analytically may not have been fully validated, for instance, for that particular API. But it doesn't necessarily have to be GMP. It just has to be well characterized. Let's see, what else was I going to? Oh, the vehicle. You know, last week I heard something. I don't know how many of you were here. Murdad, I was a little surprised that they were using a vehicle with 40% cyclodextrin. Vehicle choice is critical. I mean, critical for toxicology studies. And you can't make assumptions. You can't make an assumption. Oh, this excipient is used in a human product, so it'll be okay. No, it's approved for humans. Doesn't mean it's okay for a mouse, toxicities. I'll share one with you. It's remarkable, really. Years ago, Bill, were you with Syntex, Bill, too? You so I've told this story here before. During the development of mycophenolate mofetil, CellCept for prevention of graft rejection, originally approved for kidney, you know. So originally worked up orally. We're going to make an IV product. I'm down there at the animalry with animalry. So I had a postdoc in France, so animalry, animalry, and guys came back. They told me the dogs were developing an allergic reaction. And it turned out, and now it's known, right? It's in the known space. Turned out one of the between 20 in dogs IV, no other animal, maybe. I don't think any other animal, and other routes were fine even for dog, but dogs don't tolerate IV tween. They broke out in hives. You know, we looked at it as much as we could before doing the work, and then we went back again, looked at all the literature I could. Of course, the only paper that was available really was in German, so it was missed initially. You know, and there were not too long after that, it became a reasonably well understood and known phenomenon. But back in those days, you know, no. So the choice of vehicle and the recognition that you need to appreciate each species that you're using in terms of vehicle is tremendously important. And these days too, I hear, you know, you can formulate a lot of things, but for tox work, you don't need to make it perfect necessarily, right? We need to get an exposure that's adequate to characterize toxicity. That doesn't mean we have to make its bioavailability to 10 to 20 to 100x more than that level. I mean, I go through this a lot. So in other words, sometimes it's very simple formulation, a suspension formulation, you know, not a nice thing if you're a pharmaceutical formulator, but it works, you know, and we understand methyl cell, you know, there's no tox liability to methyl cellulose. So anyway, paying attention to the vehicle is good. You know, in there too, that reference I have, Shane Gadd, a colleague of mine, he and a couple other guys I know have published some papers over the years. I think there's a more recent one than the 2016 paper now, but it's a compilation of excipients, right? And it's a very nice starting point. It's not, you know, nothing's ever perfect, and really, but it's a very good starting point, and it's a pretty broad survey of vehicles, and it's a good. They're two good references. Let's see. Yeah, maybe just the point here on dose level selection. You know, Steve and I were talking earlier today. You know, how do you get started, right? How do you picking doses, whether they're for the tox studies, whether they're maybe for animal studies for efficacy, right? I told Steve, I mean, in my sphere, I mean, these days, early scientists do a great job. I mean, we appreciating. You know, what works, like in a dish with the relevant cells, right? And then translating that into a targeted actual plasma exposures, really, right? So they do a pretty good job. I mean, most projects I've worked on these days, I have a starting point, a target. And with that, and that's important. I mean, you want a target, you know? And with that target in hand, even in these very first studies, I like to work in a range that's 3 to 30x of the clinical of what we project as a clinical exposure. I'm going to show you in a little bit. The expectation from the FDA could be to go higher than 30x, but this is a relevant range. In other words, I can do these first studies 3 to 30x, even if nothing happens at 30x. If going to the 4-week study, I'm only increasing the dose a little bit more maybe, but the bulk of the doses I'm going to use, I'm already having some pretty good understanding of. So I'm de-risking the dose selection in this initial study. But if you underdose, right? If you're a little timid, you know, and you underdose in these early tox studies, you run tremendous risk because you're gonna ultimately now have to explore we might wind up at a considerably higher dose going forward. That's not a good move. If what you want to be doing as you go forward is limiting the dose, decreasing the dose, because from that perspective, you're now understanding what you're dealing with. As you're increasing the dose, right, you're going into the unknown territory. The other thing I like to do too, particularly when there's not a lot of information available, is to have a little gap between starting the work in a rodent, a rat, and then working in a dog or a monkey. You know, at least a little bit to appreciate the general health of the animals. So sometimes I like to have at least a week separation in the start of these studies, if not 2. It doesn't really have to be longer than that, but a little gap. I think I said this earlier. I always consider these early studies like the nonlinear phase of toxicology. You know, I've had it happen. 14 days, everything's fine. Day 15, it all falls apart. It's remarkable what can change. And actually now, when people look back, taking the surveys, you know, across, the industry and drugs, more or less, there's an agreement that the bulk of what we're gonna pick up as tox liabilities happens in 28 days. I mean, in general, you know, the greatest number of cases, I mean, there are always exceptions, you know, but certainly by the time you do a 90-day study, for a lot of, you know, and this is always depends where you are too. When I'm all the situations I'm involved with, when we talk about in-licensing a molecule, I like to have 90 days of data, even though I do appreciate 28 days is pretty revealing. But again, that this is a good point to appreciate, and that's back to that point I made earlier. You do single dose tox work to get into the clinic. You could be missing everything, frankly, everything. You know, so we did the range-finders. We get to the GLP studies. You know, they're very similar. I mean, they're larger. I mean, that's the main difference here. We're doing them as under GLP conditions. We've validated the assays for the bioanalytical. We validated the assays for measuring the stability of the formulations we're testing. But they get bigger, you know. So typically, we're using 15 rodents in these studies. 10 for the end of the treatment period evaluation. 5 go on through a recovery period for a month. For a 4-week study for a month. For the non-rodents, for studies of 28 days or up to 13 weeks, really up through 3 months, we're using 5 per sex. And these numbers, you know, this is the these are supported by and agreed to or globally. I mean, yeah, but we throw in a few more endpoints into the GLP studies. I mean, for the non-rodents, we're doing electrocardiographic, and most often electrocardiographic workups. You might notice I drew, there's some specialists that I've got listed here. For me, I'm always wanting to be working with people that are trained, you know. So there are veterinary eye doctors, right? So we talk to veterinary eye doctors, veterinary clinical pathologists. So you want to employ these folks as you go forward. How am I doing on time? Okay. Where is that clock? Right there. Okay. Okay. Again, the studies as you go forward, the primary difference here is the size of the studies, so you can get an appreciation of this. But the largest studies that we run are lastly, right, these carcinogenicity assessments become remarkably large. That's a minimum 50 animals per sex. The goal of a carcinogenicity study is to evaluate the effect of the drug over a 2-year period. So survival is what you want. You maximize survival. If you anticipate a tox liability, study size increases. So, I mean, I think more often than not, I'm running studies that are 60 animals per sex. I added some costing to this. I've updated this slide. In the past, I've had dogs on here. I didn't put them on this time, but just to emphasize the cost of using primates these days. I mean, for a 1 month study, it'd be running shortly. And these are real prices. I mean, these are from contracts signed with CROs in the United States, right? So $3 million nearly to run a 1 month monkey study in the United States. I mean, the prices are still a little bit less outside of the US. I work with Wuxi over in mainland, and the prices are less. But yeah, it's getting expensive. I think the last slide I had like this made with the dog was under $2 million. And then just to compound what I've just shown, I mean, they only get higher, right, as we go forward. So it's rather remarkable. And I'm not kidding. I really do think when I started, I mean, these were approaching the cost for an NDA, $20 million. I want, just to talk a titch about dose selection, appreciating we don't have much time. But there is a clear expectation from regulatory authorities around the world into high dose selection for toxicology studies. And this is taken from that guidance document I pointed to earlier, M3R2. But appreciate that in the goal of the studies is to elaborate, uncover potential tox liabilities. So dosing up to 1,000 and more milligram per kilogram is within the realm of expectation. Before you say, you've exhausted what could be done in an animal. The first part of this, that little box, maximum tolerated dose, right? You limit a dose based on toxicology. Exposure saturation, kind of obvious. The more you give, you don't get any more in the blood. That can be a limit. A feasible dose, you can only give an animal so much, right? In terms of volume, for instance, right? But the expectation that when in situations where you're not seeing tox is that you try to approach a factor of 50x above the targeted clinical efficacy dose, right? That's a remarkable margin. I mean, really. But anyway, to be prepared for. I don't often get caught in this type of stuff, but it happens. I mean, I've had cases where dosing up to multi-gram levels in animals, and that's a burden. Back to the guys that are making it for sure. I put this table in here. This table is also in that guidance document, but I find this table sometimes helpful in the beginning, appreciating the relative clearance differences. This was largely based on clearance differences across species in terms of helping to predict what a dose range might be when you're without much data yet. This table also helps is a conversion from the animal doses to human equivalent dose based on clearance. And it's useful, in helping establish what that first human dose is going to be. Again, that's first human dose, it's a summation of the toxicology data, but it's driven by what we consider to be the non-adverse level in animals, the most sensitive animal, in other words, the lowest non-adverse level dose. And then from there, in general, we restart in humans about tenfold below that number. Maybe just in light of time, I'll go over this. Just suffice to say, you're employing experts when you do this work, because without expertise, sometimes it's difficult to draw connect the dots. What really is adverse? What's not adverse? These are critical decisions. So there are a lot of things that happen that we consider as adaptive changes in animals. They're not toxicologies. They're physiologic changes. They're reversible. So again, there's lots written these days, and there are plenty of people out there. And most big CROs work with people that are appreciating these things. I talked the need for reproductive toxicology evaluations is there, but for compounds that are going to be used chronically. Men, a little easier time with men because the reproductive tissues can be examined histologically without functional assays, right? Histologically, and determine if there's going to be much of an issue with potential damage to reproductive organs. But for women, a lot more, particularly for women, childbearing potential. I mean, we're a lot more rigor involved. So not only are we assessing the tissues histologically in general tox studies, but now we're going to do functional evaluations on reproductive performance. And the expectation is that in many regions of the world, these studies have to be, some level of this type of evaluation has to be done before women are enrolled in clinical trials. In the United States, we have a little more latitude to work. These studies don't necessarily have to be done before phase 3. But again, this type of work is done for chronically used molecules and going into women childbearing years. Yeah, this is just some, I'll skip over that one. This, maybe just to spend a minute on this slide. Again, there's been, in general, for these chronically used molecules, we've done carcinogenicity work, we do carcinogenicity work. But these days, there is a move towards creating a weight of evidence argument as to the need of this type of work. And I'll give you an example on a molecule I'm working on. It's immunosuppressive, So the expectation is, regardless of what you see in an animal, in somebody at some time, right, there's going to be a possibility, a probability for the development of a tumor. I mean, that's just how it happens when your immune system's suppressed. So with that, right, one of the aspects of the waiver is if that's the situation, I mean, regardless of what animal testing, whether it was positive or negative, we're still going to put in the label, right, that there's a potential for a tumor development with the chronic use of this compound. But the FDA, the ICH, they've opened up the door. They gave a very nice through the amendment or addendum to the one of the ICH carcinogenicity guidances. They've just broadened the thoughts that you need to go through in order to entertain getting a waiver. So it's nice. I'm hoping, I have my fingers crossed, because I don't understand how we could not get a waiver for a molecule that's going to cause a known level of immunosuppression. But that's a new thing that's come up in these last couple of years. Maybe just real quick, more than that. So people, when you're thinking of developing a molecule for pediatric use, just to be clear, it can't go backwards. I'm sorry. Can I? Yeah, it's not going backwards. Oh, there you go. I just wanted to say the table I'm going to show next is in this guidance document. And so let me go forward now.
You can also.
Oh, right there. Oh, there we go. Okay. Hey, so this is important, this table, this guidance document. Just because you're going into a pediatric population doesn't immediately mean that you're doing pediatric animal studies. And this illustration can give you an appreciation on that. It like a lot of things we do, it depends, right? And largely what this is showing, it depends what you know as you get ready to go into a pediatric population. If you're going cold-cocked into a pediatric population without any data, we're going to do a lot of testing in animals before we do that, right? If you've have data in adults and you've kind of slowly stepped yourself down into through the ages into younger and younger patients, you have a appreciation already of what the expectations are in terms of safety, and so the path is a little less cumbersome, really. So it's what I always do and tell people is don't assume. I mean, you give it a very thorough thought evaluation, and then you make a proposal to the regulatory groups you're working with. And that's how it starts. If they don't like the proposal, they'll tell you and make you do some more. But it's again, as we've talked throughout, I mean, it depends. There's a lot of depends, you know. What do you have in terms of data, and where are the diseases you're going into? Hey, and just we talked a little bit about this earlier. There's actually a guidance out there, right? How you do this, you know, what type of tox work you do for reformulated drug products, repurposed products, you know. I just wanted to basically point to these guidances real quick. There's specific guidances in terms of tox work to be done to support oncologic drug development, guidance for cellular products, gene therapy. So there's plenty of starting spots, you know, in terms of guidances out there when you begin to consider what do I need to do to get this thing into the clinic or get this thing to approval. And that's it. Yeah. So, and I'm sorry I talked too long.
Yeah, I was wondering, the dose escalation study in phase 1, does that allow you to circumvent a lot of animal studies? Or not really?
Not really. That's a standard approach. You know what I mean? We go through a single dose escalation, right, and figure out what dose we're going to bring forward, and then go into a multi-dose situation. You know, and sometimes that multi-dose is an escalation as well. Sometimes, you know, but often, you know, we have a separation between the dose level cohorts, right, in these early studies. Yeah.
You mentioned that tolerability varies based on the type of diseases you're treating. Can you speak on which aspects are more, I guess, most universally held the highest bar versus others that may have more flexibility depending on the disease?
Yeah.
In terms of what it means for a go or no go decision.
Mm-hmm. Yeah. Well, again, the easiest example of this is when we contrast life-threatening oncology diseases, right, versus a non-life-threatening chronic diseases, right? This is where it becomes more black and white, right? So the sense of urgency, right? So the you know, typically for life-threatening, and you know, again, I should say, you know, the requirements for non-clinical safety testing for oncology products, it's changing. You know, it hasn't changed completely yet. But more and more of the life expectancies are increasing with the various therapies that have been approved. So I'm working on several oncology products where studies that we would never have done in the beginning, you know, historically, are being done. Carcinogenicity studies for post-approval. But typically, if it's life-threatening, right, we're going to go. We do the initial studies for say maybe 4-week studies to get into the clinic. Rarely do any safety pharmacology, standalone safety pharmacology studies. Maybe one in vitro gene tox test, an Ames test, and then after doing those animal studies, you can dose, particularly when the patients are showing some improvement, something's happening, right? Well beyond 28 days clinically, you know. We come back and historically, we do a 3-month tox studies to support the approval of the molecule. But the 28-day studies open the window to indefinite dosing clinically, right? Whereas at the other end of the spectrum, no, you know, you dose an animal for 2 weeks, you dose a human for 2 weeks, you dose an animal for 4 weeks, then you can dose a human for 4 weeks, you know. So that's really the basic, you know, clear separation. And in the middle, there are instances where for rare life-threatening diseases where sometimes things are requirements are lessened, but that's like case by case, regulatory agency by regulatory agency. But the appreciation for what to do for life-threatening oncology diseases versus non-life-threatening chronic diseases, that's clear, very clear. Yeah.
I'm just gonna do this.
I have a question for both of you guys. So we had our visiting scholar Tinashe talking a few weeks back, and he pointed out that 40% of patients in Southern Africa had a 2B6 mutation that prevented them from metabolizing efavirenz effectively. I mean, that's a big chunk of the population. And maybe, another significant proportion, I forget the numbers, of women in Africa would not activate tamoxifen into its active form, so it was essentially not efficacious in a huge proportion of Black women. But how would you guys propose we start to deal with this in a more proactive way instead of just throwing this out there and seeing who gets sick or which we will eventually see, but seeing who doesn't respond, we're not going to pick up, in this case, probably for decades. What are your thoughts on how we deal with this in developing therapeutics?
At the in vitro level, right? Like once you know about the genetic differences, you can set up a panel of assays, and there's a whole field of predicting pharmacokinetics from all of that data. So the computer programs or software to help do those things. But I'm by no means an expert on the fundamental things that I get involved in, figuring out the structures of things. That would happen much more at the bioanalytical, pharmacokinetics end of things.
Yeah, Kevin, I'm smiling because, so largely, I support pharma, right? And but largely here, you think outside of that box, right? So it's interesting in pharma. We don't really. There are certain markets, right? They're gone after, right? And this excludes, I think, problems that you're thinking of now, right? No justification in terms of how I'm speaking to it, but that's how it works, right? Because like Bill talked, we have the tools to appreciate these things these days. Really, we incorporate genetic evaluations in clinical trials these days all the time to try to predict to understand which populations are going to be responsive, non-responsive, So the tool and the appreciation for metabolic differences around the world in terms of phenotypes, has been known, So I think
it's not really well known, Mike. Tinashe has identified mutations in CYPs that are present in 5% of the African population that were unknown.
Yeah.
That he sort of discovered looking at metabolism of 5FU.
Yeah. It's like a sad truth, I guess, right? At the end, then it's like a underrepresented people, underserved. It's just such a multifaceted question, Kevin. Who has the ultimate responsibilities, right? So it's not something that seems, well, it hasn't been, right? So it's not so obviously addressable, right, or answerable.
Yeah, you can envision at some point that with programming, patient-derived iPS cells, for example, when you actually know what their sequence is and their enzymes, and actually making hepatocytes and microsomes and sort of looking for difference. Yeah.
That's the point. It could be done. It could be done. It's just at the point. So it's almost like not the technical side of the coin that's the problem, right?
John.
Nice talk. A quick question. wanted to get your thoughts. There are now a couple of examples of people who have used the same excipient, lipid nanoparticles, the formulations that were used in approved drugs, and have used the same derivatization of the antisense oligonucleotides, the morpholinos, the phosphorothioates. And they've gone, as far as I can tell, directly into rare diseases without doing extensive toxicity. Your thoughts about the only thing being different is the nucleic acid sequence?
Yeah. So I don't work with nucleic acids for starters, right? There, as an example of something, historically, we've had higher levels of freedom to operate when we're within a very well understood chemical class. That's historic. So there's an ability to have more freedom to operate. There always has been. That's why I alluded to, even though that law came out this year, the FDA doesn't have to require, right? They never had to require, right? So the freedom to operate is there. And maybe like drawing on examples, vaccine development, when things are well understood, there's for sure, there's the ability to move quicker, right? To limit, the amount of testing. It's also the urgency, right? I can remember those antibodies that were maybe, I don't know who's exactly went into this, but I thought it was a Gulf War, it was before then, but when the antibodies for sepsis were being developed, they went into soldiers. Prior to testing, robust testing, so there definitely are examples of freedoms to operate. They're based on having a good understanding of the basics, and basics in terms of from the chemistry to the efficacy to the liability. So for sure, I don't mean it's just in general, there are more restrictions, and certainly for new things, right? And that's the space I work in is new things. Just one more thing to add to that. I wish we didn't have to use animals. That's a great thing to get to. But I also have plenty of examples, Bill probably does too, where an atom gets changed. It's hard to believe, right? An atom gets changed, and simple atoms, hydrogens get changed, and the profiles change, the toxicity changes. So it's difficult. To think that someday we'll get away from the animal testing. But anyway, it's long-winded. But your point's well taken. There are definitely, depending upon the need and the understanding, there are examples of where we limit the animal testing.
Great, thanks.
Yeah.
Any more questions? All right, let's thank our speakers one more time. Thank you.
Oh, yeah. Oh, yeah.
Okay, yeah. Okay, no, actually leave it on, because students.
Oh, you're welcome. Yeah, sure.
Students, the slides are already on Canvas. Anybody else can ask me for the slides. And yes. And don't forget to send me your slides for Friday.
No, I think I'll only be able to.